Hello, everyone, and welcome back to our lecture on signaling transduction. I am Dr. Muzamil Liaqat Ali, Assistant Professor of Biochemistry. In part one, we thoroughly covered GPCR structure, receptor classification, and the complete cAMP protein kinase A pathway with all its steps and termination mechanism. Today, in part two, we will continue exactly from where we left off and master the remaining pathways: the cGMP pathway, the IP3 or the DAG pathway, and intracellular signaling by lipophilic hormones, right? These pathways are extremely high yield for exams or in clinical practice because they remain because they explain the mechanism of action of nitric oxide donors, Viagra, that is Sildenafil, steroid hormones, and many antihypertensive drugs. So let's proceed with the full depth and clinical correlation, right? So let's discuss the role of inositol triphosphate, the IP3, and diacylglycerol, the DAG, in intracellular signaling. We now enter the phospholipase C pathway, which is activated by GQ coupled receptors. When a ligand, the very first messenger, binds to its GQ link GPCR, the receptor undergoes a conformational change and activates the GQ proteins. The alpha subunit of GQ, that is with the GTP bounded form, right, stimulates the enzyme phospholipase C, or the PLC, located on the inner leaflet of the plasma membrane. The PLC, phospholipase C, hydrolyzes the membrane phospholipid, that is PIP2, right? That is phosphatidylinositol 4,5-bisphosphate into two powerful second messenger. The very first one is the inositol phosphate, or the triphosphate, that is IP3. Let's talk about some of the details of the IP3. That is water soluble, diffuses into the cytosol and binds to the IP3 gated calcium channels on the endoplasmic reticulum. This causes a rapid release of steroid. This causes a rapid release of stored calcium into the cytosol, dramatically raising intracellular calcium levels. The next one is the second component that is being produced by the hydrolysis of the molecule, that is PIP2, right? The very first one was the inositol triphosphate, and the second one is the DAG, diacylglycerol. That remains in the membrane, and together with the increased level of calcium activates the protein kinase C, the PKC molecule, right? The PKC, the protein kinase C, is a serine or threonine kinase that phosphorylates multiple target proteins, right? Leading to diverse responses such as smooth muscle cell contraction, platelet aggregation, and secretion of the hormones, and along with gene expression, right? So let's talk about the clinical correlation regarding this process or this step, right? Or the diacylglycerol. This pathway is critically involved in vasoconstriction, right? For example, an angiotensin 2 or the vasopressin, right? Platelet activation that is being done by the thromboxane A2, and many inflammatory responses. Drugs that block GQ coupled receptors are the alpha blockers, right? They act here, right? So remember, IP3 is equivalent to the calcium that is responsible for the calcium release, the DAG, and along with that, DAG plus calcium. So that leads to protein kinase C activation. This is a very frequently asked exam topic, right? Or the question, right? Okay. Now let's outline the different types of signaling transduction, right? The very first one is let's summarize the four major signal transduction pathway we have studied so far, right? The very first one is the cAMP pathway that is directly linked with the GS protein. So rapid amplification via the protein kinase A, right? The next one is the cGMP that we are going to discuss in this lecture, right? So cGMP pathway that is directly linked with the guanylyl cyclase that is responsible for the smooth muscle relaxation and vasodilation. The very next one is the phospholipase C pathway that is linked with the GQ protein, right? The IP3 or the DAG or the calcium or the protein kinase C activation process is directly involved in this process. So calcium signaling and protein phosphorylation are the main mode of action or the mechanism or through which this mode of signal transduction occurs, right? So next one is intracellular receptor pathway that is linked with the gene transcription used by the lipophilic hormones. So understanding these four pathways allow you to explain almost every single hormonal and pharmacological effect in the body. So draw this summary in your notes. So it is extremely high yield. So ligand, then that is responsible for the binding with the receptor or at the receptor, adenylyl cyclase, cAMP, and that is directly responsible for the activation of the protein kinase A. That is directly linked with the cAMP pathway that we have already covered, right? So PKA, that is protein kinase A. This is the mode of action through which the very first pathway, the cAMP pathway, acts, right? The next one is the ligand, the receptor, the phospholipase C, that is directly responsible for the conversion into the DAG and IP3 that ultimately leads to the activation of the protein kinase C and the calcium release, through which the cGMP pathway acts, right? The next one is the ligand, receptor, guanylyl cyclase, the cGMP or the protein kinase G pathway, right? So pay special attention to the third pathway today because it has a direct therapeutic amplification or application in cardiology and urology, right? Okay. So cGMP pathway. This pathway is unique because the receptor itself is often the enzyme guanylyl cyclase, the GC molecule. There are two forms, right? The very first one is the membrane-bounded. So membrane-bounded GC, that is activated by the atrial natriuretic peptide, and ultimately the ANP or the atriopeptins, right? The second one is the soluble guanylyl cyclase. So that is activated by the nitric oxide or the NO molecule, right? So let's discuss the step-by-step mechanism, right? The very first one is the ligand. The molecule, as we have already discussed, that is ANP, right? The very first molecule is the ligand is the very first molecule that is the primary messenger, right? So ANP or the nitric oxide is the primary messenger, binds to the guanylyl cyclase. The next step is the activation of the GC molecule. Guanylyl cyclase converts the GTP molecule to the cGMP molecule and producing the cGMP along with the PPI, right? Pyrophosphate molecule, right? The next step is the cGMP binds to and activates the protein kinase G, right? The PKG molecule. And then the next step comes, right, into play. The PKG phosphorylates specific target proteins in smooth muscle cells. And ultimately, the phosphorylation occurs, and that leads to either the decreased intracellular calcium and the dephosphorylation of the myosin light chain molecule, right? And the third mode, or that the phosphorylation leads to relaxation of the smooth muscle. Ultimately, that leads to vasodilation, right? So let's discuss the physiological or the biochemical role of the ANP, right? At this very step, right? ANP released from the cardiac atria when blood volume increases, causes natriuresis, diuresis, or the vasodilation, along with the inhibition of the aldosterone and the renal release, a natural defense against hypertension and the volume overload. Now, the nitric oxide that is being produced by the endothelial cells in the classic endothelium-derived relaxing factor and maintains the basal vascular tone, right? So, what about the termination? Termination is basically the cGMP is rapidly hydrolyzed by the phosphodiesterases, especially the PDE5 in certain tissues back to GMP, right? Now, the clinical application, right? Clinical application, the Sildenafil or the Viagra and the PDE5 inhibitors, right? This is one of the most important clinical correlation in the entire lecture, right? In corpus cavernosum, right? And pulmonary vasculature, PDE5, right? PDE5 is the main enzyme that breaks down cGMP. Sildenafil or the Viagra or tadalafil, right? Or the vardenafil or selective PDE5 inhibitors. They block the degradation of the cGMP. That leads to sustained or high levels of cGMP. That leads to prolonged activation of the PKG molecule, and that leads to prolonged smooth muscle relaxation and ultimately causes the vasodilation, and that leads to erection, right? In erectile dysfunction, this allows increased blood flow into the corpora cavernosa when the nitric oxide is released during sexual stimulation. In pulmonary atrial hypertension, the same mechanism reduces pulmonary vascular resistance, right? So here, the exam tip for you guys, right? Always remember, nitric oxide that leads to the GC molecule, guanylyl cyclase, that ultimately goes to the cGMP, and that stimulates the PKG molecule, right? And that leads to smooth muscle relaxation. So PDE5, right, inhibitors prolong this effect. This mechanism appears repeatedly in pharmacological, biochemical, or clinical, questions that are of high yield in every single exam, right, related to cGMP pathway. So let's discuss the signal transduction via the intracellular receptors, right? So now we move to the fourth major pathway used in the lipophilic hormones or the lipid-soluble hormone, steroid hormones like the cortisol, aldosterone, estrogen, progesterone, or the testosterone, thyroxine, the T3, the T4, vitamin D, calcitriol, or retinoic acid can freely diffuse across the plasma membrane because they are hydrophobic in nature. Inside the cell, they bind to the intracellular receptor located in the cytosol or already bound to DNA in the nucleus, right? So the hormone receptor complex undergoes a conformational change, exposes the DNA binding domains, dimerizes, right, and binds to specific DNA sequence called hormone response elements, the HRE, right? So this leads to the recruitment of co-activators or co-receptor repressors, and ultimately increases or decreases the transcription of a specific gene. So that leads to synthesis or repression of a new protein. So what about the important structural details right now we need to discuss? Steroid hormone receptors have 4 domains, right? The very first one is the variable N-terminal domain. That is responsible for the transactivation, right? The second domain is the DNA binding domain. That is zinc finger motif, right? The third one is the hinge region. And the fourth one is the ligand binding domain. That is C-terminal. So what about the thyroid hormone, or why the thyroid? Let's have a special note for the thyroid hormone, right? Its receptor is already bound to DNA even in the absence of a hormone, binding to the T3 that converts the receptor from a repressor to an activator. So what about the clinical correlation at this very step, right? So this genomic mechanism This genomic mechanism explains why steroid hormones have delayed but long-lasting effects that may prolong from hours to days, right? Unlike the rapid non-genomic effects of the peptide hormones, right? So drugs such as tamoxifen, that is estrogen receptor modulator, and spironolactone, that is aldosterone receptor modulator, right? Antagonist, act exactly at this level. This pathway is central to endocrinology and oncology. So we have covered all four major signal transduction pathways in complete detail: cAMP, cGMP, IP₃-DAG, and the intracellular receptor pathways. So, dear students, we have successfully completed the entire lecture on the signal transduction. In part 1, you mastered the GPCR structure and the cAMP pathway. In part 2, or in this very lecture, you have gained deep understanding of the cGMP pathway with Viagra mechanism, the IP₃-DAG, that is calcium signaling pathway, and the intracellular genomic signaling used by the steroid and the thyroid hormone. These four pathways form the molecular foundation of the virtually every single hormone, neurotransmitter, and the drug action you will study in pharmacology or the medicine, right? So draw each pathway at least 2 to 3 times, compare them side by side, and link them to clinical conditions. This is the best way to retain these pathways permanently. Thank you very much for watching both parts of this lecture series. And these lectures, if these lectures are helping you, please like both videos, share them with your batchmates and juniors, and subscribe to the channel. Turn on the notification bell for so that you never miss the next high-yield lecture. Please write any doubts, especially about the cGMP pathway, Viagra mechanism, IP₃-DAG pathway, and or steroid hormone receptor in the comment section. I personally read and reply to every single comment to clear your concept. This is Dr. Muzamil Liaqat Ali signing off from this lecture. Revise all your 4 pathways thoroughly, correlate them with clinical pharmacology, and stay consistent in your studies. I look forward to seeing you in the next lecture. Thank you very much, and keep learning biochemistry with passion, precision, and clinical insight. Thank you once again.